Now we have Matthias Karl, and Matthias joined recently Cibio, and he will be happy to tell us what are the reasons that he decided to move to Cibio, and what is his path in his career.
Yes, hello everyone. Good morning. Well, I'm actually a trained microbiologist who did the master thesis then a bit later, that's already 25 years ago, in virology back at the University of Göttingen in Germany. And then I moved to the Max Planck Institute there because there was my professor at the time who really hooked me onto an animal model, that is the fish animal model. And he showed me how he can make this animal produce a part of the eye structure instead of an ear. And that fascinated me so much because before I was only seeing cryptic bands on gels and statistics that I stayed with a fish and with neurobiology, and that's what I'm also a neuroscientist. I moved then to the EMBL in Heidelberg, one of the best places to do research in Germany at least. And after that, I did a postdoc time at the University College in London. Then went back to Heidelberg to the university in 2009, and finally I came here to Cibio about 3 years ago at the end of 2017. And the decision to come here was relatively easy because as so many researchers, I'm also an inbred researcher. I'm married to another scientist. And as you might have heard already, it is really hard for a scientific couple actually to find a place where we can actually have a normal relationship with a family together in one house instead of commuting like so many other research couples do that. And Cibio is a fantastic place in Italy to do research, still alongside doing a lot of teaching work, of course. And it's also a very young university and department. Also, with my 52 years, I'm one of the oldest here, if not the oldest, in our institute. So we are very interactive and very nicely equipped also. So I think this is really a good place. And on top of all these advantages, the location is of course also great because as a biologist, I'm very much into nature, and the Trentino area is of course a great place to live in.
True, very true. So Matthias, you were telling us before that you become fascinated by neurobiology by working with this animal model, with this zebrafish, your animal model. Do you want to tell us what you do with it and what is your, let's say, an example of your research day and goal in your days and so on, just to give an idea.
Sure, of course. Actually, there are several fish animal models out there, and I started off with another fish that was a Medaka fish that is originating from Japan, and now here at Cibio, and also before in England, I came to the zebra fish, and we are studying brain development. And one big advantage of this animal model is that it is totally transparent, so you can see anything that happens during development under normal conditions or diseased conditions in the living embryo. And that is absolutely fascinating on one side, but also on the other side, this is very, very helpful if you want to see what, for instance, gene function is in vivo, so under physiological conditions. And many of the genes, more than 80% of the genes that we have found in humans to be mutated and cause diseases are actually evolutionarily conserved. So with this relatively cheap animal model, we can really find then also therapies and cures for these human diseases. We are currently actually doing also, for instance, a compound screen for drugs that interfere with a neural circuit that is very much affected in human patients that suffer from depression and schizophrenia and also autism. And we are onto some compounds that might, in the future at least, help these patients then in reality, sort of. What was your other question?
The other question, if you want to describe us, what is your daily
my daily life. Well, in non-COVID conditions, my daily life is sort of being in the lab around at 9 o'clock and stay there until the evening, until 7 maybe, go home, have dinner, and then start again at around 11 when the family is in bed to keep on with my work. And as a group leader with a few people that work for me in the laboratory, I'm of course supervising them and see what they are doing, and have meetings with them to tell them what I want them to do. And on the other hand, I'm mostly at the computer trying to find funding for our research, to evaluate other people's work, review papers and grant applications, this sort of thing. And of course, more than half of the time is dedicated to teaching. So we're, of course, a teaching university here, and we have to make the students happy and learn something about research and biological questions. In my case, I'm teaching mostly developmental biology. This is my field of interest also. And I'm trying to also transfer some of my experiences in doing that job.
Yeah, that's very true. That's very true. So related to this, I wanted to ask you a question that maybe is more for the background of the students of this master, which is about, so what do you think that the public opinion should know about an important discovery, and how do you think this should be communicated to the public?
The public should certainly know what we are doing, and especially when we discover something great. that is of course very, very important also for their attitude towards research and how important that is. I mean, we have currently the situation here with the COVID virus. That's the prime example on why research is actually very important. And people should especially know, I mean, from my point of view, since I'm from a field of basic research, that basic research also using animal models is pivotal for discoveries like we have now, the discovery of a vaccine. And to transfer this information to the public is, of course, very important also with respect to our funding situation, that they also understand where their tax money goes, in part at least, and that they can comprehend that without research, we are much worse off, obviously. So we are not only playing around like some people would say if they hear, oh, this researcher, this professor is doing something with Drosophila or with zebrafish. What the hell would this help us? I mean, they really need to understand that what we are doing is actually saving lives. And that's why I think also your job as science communicators is very, very important.
Sure. That's why actually this is interviews are for the students to maybe have a much touch feeling to what we do in reality. And so one last question is, what is your suggestion? Maybe you already said it a little bit, to the students of this course. So what you recommend to them?
Well, keep on going. We depend on you. That is really a very important job that you are doing. Most of us researchers, we are sort of a bit brain dead, such that we, in our little world of research, we think much too complicated most of the time, at least for myself. So my personal test of if I'm able to communicate science is actually my mom, who's more than 80 years old. And if I'm able to make her understand what I'm doing with my fish, then this is a good control for me that I managed to explain it in a simple way. But I'm certainly not well suited to do that. Although, of course, we are also doing press releases, but we are also helped by people like you to make ourselves understood. So please go for it.
Thank you. Thank you, Matthias, for the kind interview. And then if you like, the 28th, we will gather together for the class. I will tell you what time, I don't know yet. And so you can maybe answer to some of the questions that the students may have.
Thank you for having me. Thank you.
Thank you very much. Bye.